When conducting a statistical study, planning is key, and one of the most important factors is you know, getting the sample, the method of putting together the sample, and we're going to look at that and a few other things in this section 1.3, and we're going to talk about what a simple random sample is, and if we do not collect data in an appropriate way, then basically the data is useless because bias or any of the other pitfalls can mess up your results. So if we want good, honest results, we really need to keep bias out of the picture and to try our best to get a random sample. But first, let's look at something that's called the gold standard, and it really is. And when I see this, I typically think about medical studies because that's where they use this the most. Pretty much exclusively, they use this placebo slash treatment sort of setup. And a placebo is basically just a sugar pill, right? It's a pill that, of course, the person taking it in the study, they don't know whether they're on the placebo or not. But so it like an example would be where someone, maybe studying a new pill for blood pressure or a heart pill or, whatever, just some kind of a treatment. And in order to assess whether that treatment is effective, then they need to be able to control, who is on the medication and who is not. And of course, typically nobody knows whether they're on the placebo or not. And we're going to look at that in a second. That's called blinding. And we're going to look at two different types of studies. We have observational studies and experiments. And my example about the medical study, blood pressure, checking out, studying blood pressure medication or whatever, that would be an example of an experiment. So with an experiment, we apply some sort of treatment and then proceed to see whether or what its effect is on individuals. Does it do any good? Was there a significant reduction in blood pressure, among those who actually got the medication and stuff like that. Oh, another thing are these terms, experimental units and then the subjects. So the people in the study collectively are called the subjects, but then individually they're called experimental units. So let's also look at an observational study, and that's really just where, I always think about people going out and observing, maybe animals in their natural habitat and things like that. Like in my example, if you're in Africa or somewhere where there's some exotic animals or whatever, and you want to observe their behavior or whatever in their natural habitat, well, just the fact that you're there changes things, right? So that's, I think that's called the Hawthorne effect. And then there are others. So here we have an example of ice cream and drownings. And of course, this is designed to reveal or to illuminate a mistake here, because certainly ice cream does not cause drownings. But based on data, someone could be led to conclude that if they're only considering the data. But of course, there's a lurking variable here, and that is the temperature. So the idea is that, people usually eat more ice cream during the summertime when it's warm. And what else do people do when it's warm? Well, they go swimming. And so that's the connection between the two. But that doesn't mean there's a causation there. One doesn't cause the other. We could even conduct an experiment in this situation and have one group that receives ice cream and another one that doesn't. And I think we would quickly see that the rate of drowning victims is not affected by ice cream consumption. Replication is the repetition of an experiment on more than one individual. Well, that just makes good sense. If you have several participants in the study, you're going to give each one the treatment or whatever. And so, in order to make good use of that, requires sample size is large enough. Blinding, I talked about this earlier. So blinding is the situation where the subject doesn't know whether they're receiving the treatment or a placebo, the sugar pill. And often you hear the word or the term double blinding. And what that refers to is that the subjects in the study don't know whether they're receiving the actual treatment. And also the people administering those treatments or the placebo, they don't know. They don't, whoever's working, only certain people know, who's like only certain people conducting the trial know who receives the treatment and who receives the placebo. But like most people that are just working and administering the treatment or the study, they don't know whether they're actually giving the treatment or a placebo. And so that's here on this slide. Double blinding is the subject and the experimenter, neither one knows who's receiving the treatment. And then randomization refers to how we're going to assign subjects to the different groups. So who's going to be in the treatment group and who's going to receive the placebo. And of course, this would be one situation where you clearly do not want bias in assigning subjects to one group or the other. So that clearly needs to be done in a random way. Okay, so let's talk about what a simple random sample is, because it is absolutely critical that in a study that a simple random sample is used. So a sample is random if the following is true. So let's say we're talking about a sample of size 50. So we want to put together a sample of size 50 from our population. And what needs to be true if the sample that is selected is a simple random sample is that the probability that this particular sample that we select gets selected needs to be exactly the same as the probability that any sample of size 50 would be. In other words, the probabilities need to be exactly the same for any subset of size 50. And if our selection was done so that that's true, then we have a simple random sample. And we have no bias. So let me say that one more time. If any subset of the population of size 50 has the same chance or had the same chance of being selected as the one that was actually selected, then we've selected or we've constructed our sample so that it is random. It is a simple random sample. And okay, so let's look at some other sampling methods. Systematic sampling would be what you see there, where there's some sort of way to order the elements of the population, and then you select in this case, every third one. So the third one and the sixth one. Convenient sampling is probably not the best. That would be like, where you're just using data that's easy to get. And that's not good. It has, it's like voluntary response sampling. That's also not a good way to do things. Stratified sampling is interesting. So there are two different sampling methods where we basically divide up the population into groups, and then we select our sample in accordance with that. But so let me talk about the difference between stratified sampling and something called cluster sampling, because in both of these sampling methodologies, like I said, the population is divided into subsets, separate subsets. But the key is that with cluster sampling, let's do that one first. With cluster sampling, once we've divided the population into the several groups or blocks, if you want to call them that, then we randomly select certain of those clusters. That's what they're called, clusters. And then, like, so for example, here, these that are highlighted, those clusters were randomly selected. And once those clusters are selected, every person or unit or whatever, like all of this cluster is used in the sample, and all of this one and all of this one. So with cluster sampling, either everybody in the cluster is selected or everybody in the cluster is not selected. With stratified sampling, it's a little bit different, and you can see the picture there. Here they've divided men and women, like so they've split up the population based on gender. Now, the reason why you would want to do something like this is if you want to make sure that members of certain groups, here we have men and women, but there are several different ways that you could divide the population into subgroups or strata, as they're called here, so that we make sure that we sample a few from every one of those subgroups. So with stratified sampling, we're not going to necessarily, we're certainly not going to select all the men, and we're not going to select all the women. We're going to select some of those from each group, each subgroup. So we're going to basically divide the population into strata and then sample from each strata. So we'll sample some of the men and some of the women and put them together, and that'll be our sample. And a multi-stage sampling would be if you had some sort of combination of the methods that we've talked about. Okay, with observational studies, the key is in the name, observational. So in that situation, you're just there to observe. And like I talked about earlier, we don't want to, we want to make every effort to mitigate against any possible effects, any possible Hawthorne effect, right? It was called the Hawthorne effect. We want to try to mitigate against that. So we don't want to affect the objects that are being observed. All right, types of observational studies. So there's three. Well, we're not going to do much with this, but I think it's interesting to look through these. So the cross-sectional study is when you observe data at one point in time. So really the key here to understanding these three types of studies is time. So retrospective study is when we collect data from the past using historical records, things like that, past video clips or, whatever. Just look into the past and collect data from, there. And then, so that's retrospective. And then prospective is where we look into the future. And so we come up with some sort of method to collect data from future groups that share common factors here called cohorts. And I think for our purposes, we'll stop there. But I would suggest that you just read through this section. There's some good information there. Not a lot, not heavy on the math, just a lot of terminology. Well, and one last thing, let's talk about sampling errors. So the idea, again, with sampling, to get a random sample, is that we want to construct a sample in such a way that there's no bias, right? The sample that we have is had the same chance of being selected as any other sample of that size. And so now let's talk about sampling error. So the idea with random sampling is that there are a whole lot more samples of size 50, let's just stick with size 50, that will give us results that do mimic the population, as opposed to samples of size 50 that don't. And so if we randomly select our sample, then we'll have a good chance of getting a representative sample, because that's what we want. We want a sample that has similar characteristics to the population. But, if that doesn't happen, I mean, it could be. So let's just say that maybe 98% of all samples of size 50 from our population would be representative, and maybe 2% would not. Or, maybe it's 95% and 5% or something like that. So let's just say that, so if we do things in a random way, then we would have like a 95% chance of getting a sample that really does represent the population well. But, it could be that we just, had bad luck and we did everything right, and we didn't have bias, but we just got a sample, we just landed in that 5% or 2% where we just got a sample that was not representative of the population. And if that happens, if we have some sort of error because of the sample that we got just wasn't the best, then that's a sampling error. So that means we did get a random sample, but it just so happens that we got a sample that led us astray. So that's one type of error, is random sampling error. Non-sampling error is like, if somebody enters data wrong, or, data entry or computational error, or maybe something wasn't measured correctly, something like that. And then the third kind of sampling error would be non-random sampling error. And I think this would be the one where probably there was some bias injected into the sampling construction. Because basically with this last type, that means that there was some method employed that was not random. So like I said, in that case, there's going to be some sort of bias. Because remember, we talked about before, with a voluntary response sample, usually it's people that have strong feelings one way or the other, who volunteer, their time to provide what they think, or take the survey or whatever. But then also convenient sampling can also be a big source of bias.